As we continue talking about viruses, we can shift from bacteriophages onto animal viruses. And there are two key variables that we use to classify viruses that infect animals. Are they DNA or RNA? And then are they single-stranded or double-stranded? And you have to understand, this is of particular interest to us, humans, because we are animals. And so what we're going to do is we're actually going to dive through this little chart here. And I say little and it's relatively large, but basically these animal viruses fall into one of 6 different categories. You can see those categories here. Category 1, 2, 3, 4, 5, and 6. And we're going to go through each one of these and talk about how we classify them. Now understand, right out from the beginning, we are not talking about how dangerous these viruses are. It's not like class 1 is the most dangerous and class 6 is not. That is not anything to do with this classification system. It is simply looking at their structure. We classify viruses by structure. So no matter what class they are, 1 through 6, any of these can be absolutely dangerous viruses to us. And so we just focus on the structure themselves. So if we look at class 1 viruses, well, great example is the adenovirus, some respiratory viruses. They are double-stranded DNA. So all of these are going to have a capsid. Every virus has a capsid, and every virus has some kind of nucleic acid. But in the case of class 1 animal viruses, they have double-stranded nucleic acid inside there. So double-stranded DNA. Now you will notice in this column, not every one of these has an envelope. For example, adenovirus does not have an envelope, but the pox virus, such as smallpox or cowpox, does have an envelope. And the herpes viruses, that's herpes simplex 1 and 2, and all those, they have an envelope. So how do we classify that? Remember, the classification is based on the type of DNA, not on whether or not they've got an envelope. So class 1, double-stranded DNA. Class 2 is single-stranded DNA. And so here, this is an example of that. It causes a mild rash. And single-stranded DNA is a little bit less common. So we have lots of double-stranded DNA, not much single-stranded DNA. By the time we get into class 3, we've moved on to RNA viruses. And the first one is a reovirus here, like a rotavirus that causes diarrhea, or Colorado tick fever. And so that's a double-stranded RNA. Now, how can RNA be double-stranded? We talked about it's mostly single-stranded. Well, yes, but when it folds back on itself, it becomes double-stranded RNA. And so, again, still has a capsid, no envelope in this particular case, but class 3 are double-stranded RNA. As we move into class 4, these are single-stranded RNA viruses. Now, in this, we start splitting hairs because there's 2 different types of single-stranded RNA viruses. One, where the RNA itself is going to serve as the messenger RNA. And I hope you can understand how quickly these would work, because as soon as that RNA gets in there, it's immediately ready to make proteins. It's immediately ready to get copied, and there's no extra step involved. So one of the big ones you notice on this list are coronaviruses. So if you look here, here's coronavirus. They do have an envelope. Remember, having an envelope does not affect what class we put these in. And this is severe acute respiratory syndrome, SARS. That's what coronavirus is. Now, this particular chart was made before COVID-19 pandemic, so it's a little bit played down here. But they were a class 4 single-stranded RNA virus that used their RNA as a messenger RNA. Then we have class 5 viruses. And so these are also single-stranded RNA, but they're going to be a template for RNA synthesis. So what that means is there's an extra step. The mRNA comes in and it has to be turned into messenger RNA. There's an extra step in there. And the most common one that most people know about is the influenza virus here. It has an envelope, and you'll see all 4 examples of here have an envelope. But once again, having an envelope is not important to our classification system. These are class 5 because they have single-stranded RNA that acts as a template for messenger RNA. Ebola is another common, as well as measles and rabies. These class 5s are pretty common. The last category we often call, we just call retroviruses, but the last category is bigger than that. It's class 6, and this is where the single-stranded RNA is actually a template for DNA. And so retroviruses are the example we talk about. That's human and immunodeficiency virus, HIV and AIDS. Retroviruses take their RNA, create DNA, put their DNA inside your genome, and then their infection pathway starts from there. So these 6 different categories are all based on what type of nucleic acid they have and how their nucleic acid is actually used. So many of these viruses that infect animals have a membranous envelope, and that envelope actually helps them bind to surface molecules in the host cell and gain entry. So some viral envelopes are formed from the host plasma membrane, and some are kind of a mix. But either way, the viral envelopes are really important to allowing the virus to get into the cell. Other viral membranes will actually form from host nuclear envelope and are replaced by an envelope made from the Golgi apparatus that can include some viral parts as well. So this is showing you an envelope with glycoproteins that are going to help it get in. And then we see the viral genome here. Notice that it's acting as a template, right, to make messenger RNA. So since it's acting as a template to make messenger RNA, you should know which classification that is. That's right, it's class 5 because it's acting as a template for messenger RNA. And so now that the mRNA is made, they're going to make capsid proteins. The ER actually is creating some of these glycoproteins for it. They've completely hijacked the system, and they're going to shoot out new viruses. The broadest variety of RNA genomes is found in viruses that affect animals. So retroviruses use reverse transcriptase to copy their RNA into DNA. And in fact, reverse transcriptase is one of the exceptions to the central dogma of genetics that we see. And so we've done quite a bit of study in science on HIV and AIDS. And so here is an artist rendition of what HIV/AIDS looks like. It has a glycoprotein-rich membranous envelope that helps it get into the host cell here. And so in this case, it's a host white blood cell. That's what they infect. You can actually see the microscope images of them infecting up at the top. And so when that happens, the capsid opens up. We see reverse transcriptase is present there. We see those capsid proteins are present there. Reverse transcriptase is going to make DNA. That DNA gets incorporated into the nucleus of the host. And now it's actually going to spit out RNAs from there. These RNAs are going to assemble proteins. These RNAs are going to make those glycoproteins. And they're going to make reverse transcriptase on top of that. And once that's all done, they'll spontaneously form new viruses. And as you can see here in the images, those new viruses actually push their way out of the cell. So HIV/AIDS is one of the most iconic viruses that we talk about. And so most time people will get confused because they think all viruses work this way. But in reality, HIV/AIDS is a class 6 virus. It has single-stranded RNA that acts as a template for DNA.